Hello, my name is Masume Mohandesi. I'm a new visiting student in WPI. My advisor is Professor Timko. In this presentation, I'm going to present one of my experiment of PhD thesis, which was about biogas reforming reaction using biotemplated nanoporous nickel-based alumina. I just like to give a brief introduction about biomass and sustainability. Biomass energy comes from various feedstock sources categorized in four groups. Biomass has many benefits. The first one is that it can be depleted like fossil fuels with an abundance of plants on Earth. Biomass could be a primary source of renewable energy that's used as a sustainable alternative to fossil fuels. Burning of fossil fuels released carbon dioxide and other greenhouse gases, trapping it in the atmosphere. This fuel source has been responsible for dangerous impacts on the environment, air and water pollution to global warming. As you can see from the figure, from 1940 to 2021, the greenhouse gases emission has increased almost 7 times, which we need to solve it. But how can we decrease such gases? One way is dry reforming or biogas reforming reaction. This is a reaction between methane and carbon dioxide to produce carbon monoxide and hydrogen. But it is not an only reaction happen in the reactor. We had three other side reactions which we need to control these reactions with suitable catalyst. So we were looking for a suitable catalyst to control these side reactions. For this experiment, we used nickel as an active metal and alumina as a support. But this catalyst has two big disadvantages, sintering and coke deposition, which we needed to solve it. So our experiment was the investigation of the effect of biotemplate on the catalytic performance of nickel-based alumina in dry reforming reaction. In this experiment, we used biomass sources of plant as a biotemplate. Because biomass found in the nature has a unique structure which we can use in our catalyst to improve the structure and porosity of our catalyst. The total goal was maximizing methane and carbon dioxide conversions and hydrogen yield over dry reforming reaction. To choose the biotemplate, we took SEM images from different biomass. You can see two examples of them. The first image is related to pistachio skin, which had a dense structure without any porosity, and the second one is related to eggplant skin, which had a tube structure and some hexagonal shapes on it. So we choose eggplant skin as a biotemplate. For this experiment, we synthesized three catalysts with different contents of nickel and one sample without biotemplate to compare their results. In this slide, you can see the result of BT, which was so important because it showed the effect of biotemplate on the porosity. As you can see, we had wide range of pores distribution for the sample without biotemplate. But for the samples with biotemplate, we had narrow pore distribution, which showed the effect of biotemplate on the catalyst. Here there are the SEM images of nickel-based alumina in the present and absence of biotemplate. The SEM images A, B, C are related to the sample without biotemplate, and you can see that the catalyst had the tight and needle structure. But when we added the biotemplate, we had a spherical and porous structure. These figures show the results of biogas reforming reaction. The purple and red lines related to 30% and 20% nickel-based alumina with biotemplate. which stayed at higher levels rather than the sample without biotemplate. So we can conclude that using biotemplate had a significant effect on the activity of catalyst. So we concluded that added biotemplate could improve the structure and pore size distribution of nickel, increasing the dispersion of nickel particles and achieve higher catalytic performance rather than the sample without biotemplate for the dry reforming reaction. Thank you for your attention.